our recent research about how this sulfated dietary fiber from seaweed that can modulate the antibiotic effects on gut microbiome. So let me start the problem. So as we know, the antibiotics are saving lives to treat bacterial infections, but it also induce like unintended impact on our gut or residential gut communities, and that it can cause the dysbiosis and social disease. So the question we have is how we can mitigate this collateral damage without introducing new problems. So as we know that our diet also changes a lot on our gut microbiome. So our initial hypothesis is that whether some dietary substrates could help the bacteria to help them from the antibiotics. So to test this hypothesis, we designed a very straightforward experiment. We exposed the E. coli cells to kanamycin, one of the antibiotic, at the minimum inhibitory concentration, so the cells cannot grow, or at the MIC level. And then we supplemented the growth media with different type of carbohydrates, and surprisingly, we found about 9 of them, 9 of the sulfated polysaccharides that can even enable the bacterial cells to grow at the inhibitory concentrations of antibiotics. And we verified this experiment using the susceptibility test on the agar plates. We also tested with higher concentrations of antibiotics, and all of them shows consistent results that these sulfated polysaccharides can enable the bacterial cells to grow. So the next we asked how broad this kind of protection can be. So we kind of screened about 40 different carbohydrates, and including polysaccharide, monosaccharide, and oligosaccharide mostly. And against 8 different antibiotic classes and 3 E. coli isolates, including 2 clinical isolates from the IBD patients. So as you can see from the heat map, the redder meaning higher bacterial growth in those antibiotics. So consistently, we found those which I added a little star here. Those are the sulfated polysaccharides, specifically like Fucoidan, that have the very consistent protection of those E. coli cells. So a little bit background about the Fucoidan. Fucoidan is a sulfated polysaccharides from brown seaweed. If you walk on the beaches, you probably already see them. And it's also like in Asian food, it's a very common diet, and it's actually tastes pretty good. My favorite is this, like it's simple cooking. You add some spicy chili and olive oil. It's actually very tasty. Okay, so the next question we ask is, what's the mechanism that this kind of protection is? So there are several hypotheses. One of them is like whether maybe the Fucoidan changes how the bacterial cells respond to the antibiotic stress, and we tested that with RNA-seq and showed Fucoidan itself is barely introduced like altered the gene expression level. And then the other hypothesis is Fucoidan is a very large molecule that is not that easily to go into the cell. So we hypothesize that maybe this antibiotic can bind to this Fucoidan, and we test this hypothesis with the mass spec, and the mass spec results show, like if you can see from the right side, is if there's only purely antibiotic, we can see the signal here. But when you're adding the Fucoidan, different type of Fucoidan or different concentrations, the Fucoidan, the kanamycin signal is gone. So that's indicating that the Fucoidan can bind to the antibiotics and prevent them in entering into the cell. So we also tested this Fucoidan function using fecal communities from 2 healthy donors, and we cultured the stool sample and add antibiotics as well as Fucoidan. So as you can see from the A panel, like this Fucoidan addition actually enable the bacterial cultures to grow at a similar level to the control. And we then perform the 16S umbilical sequencing to track this community structure. And from the B panel, you see they're more closely resemble to the control group. And the next is we tested the Fucoidan function using mice, and there are 4 groups. We Fucoidan only, and kanamycin only, Fucoidan and kanamycin, and as well as control group. So we used there are 3 phases: pre-treatment, treatment period, and recovery period. What we found here is, so we found after the antibiotics early treatment stage, both of the communities. Yes, please. Well, sorry, I was in the chat. So the conclusion is Fucoidan after the antibiotic treatment, both of the abundance goes down, but the group with the Fucoidan goes at less extent, and the group with Fucoidan can recover. Some bacteria in the group of Fucoidan can recover faster compared to the control group, which and compared to the kanamycin group. So then let's go and jump to the summary section. So we found this sulfated polysaccharide, specifically Fucoidan, have a kind of broad protection to gut bacteria, and we tested it in vitro and ex vivo in fecal communities, and also see in mice specimen we see faster growth and recovery after the antibiotic treatment. And of course, this work I would like to be thankful to our colleagues who are working on this project, especially my mentor when I was a postdoc, Professor Eric Elm at MIT, and he's very great mentor and also supporting this project, and my current lab members at the UT, and also the funding support from NSF and IH. And with that, I'm happy to take questions.